Hi, my name's Lisa Keith. I'm a research plant pathologist with USDA-ARS in Hilo. We're also referred to as PBARC. And today I'll be giving a talk about managing coffee leaf rust and a research update. So the talk today I've organized into three main sections. I'll go over our fungicide testing. I'll talk about our variety testing and end with an update on all of our natural enemy research. And you can read the disclaimer below about mentioning any product or vendor does not exclude other products or vendors that may also be suitable. Before I actually begin the talk, I wanted to just give a great acknowledgement, a shout out to my amazing team in the lab, also at our center, the collaboration with the university, with SHAC, with HARC, and we definitely couldn't be doing this research without the support of the growers and producers. So for that, thank you very much. I just wanted to remind everyone about the disease coffee leaf rust caused by the pathogen Hamakua vastatrix, which produce abundant spores, which start infections. They develop into spots that produce more spores. And the disease cycle actually varies from season to season, mainly depending on the amount of rainfall. This picture shows the spores that infect through the open stomates, which occur on the bottom side of the coffee leaf, which is important to note when you're trying to control it. The fungus actually gets inside the leaf, produces mycelium, specialized feeding structures. It actually then produces visible spots where the mycelium exits the stomate, starts to produce urea, more spores, abundant spores, and the cycle continues for many cycles throughout the growing season. Okay, so for the first topic, our fungicide testing. And we start by looking at products in the laboratory to try to see the efficacy of these products against the spores themselves to see if they'll inhibit germination. And the quick method, we have spores in a tube, we mix it with a product. We actually plate that on water agar, which is just a clear media in a plate. The plates are incubated, 22°C in the dark, for 12 to 15 hours, and then we count the number of germinated spores. Okay, so this is a busy slide, but I'll try to simplify. On the X or the horizontal axis is the number of products that we've looked at, and the Y or vertical axis is actually the germination rates of these spores, and we always compare it to a water control, which you can see mostly to the right with that fancy blue rectangle. Now, overall, with the number of tests we've done, the average germination rate, if you combine all samples from all areas that we've collected spores from throughout the calendar year, the average rate is 55% germination for the samples, but it does range from as low as 32% to as high as 72%. And this shows the results of how these products affect, again, spore viability. Okay, so do they kill them in the lab, in vitro, or not? And I've tried to group everything based on colors. Now, the yellows are the systemic fungicides and Preaxors included. The blue are the copper products, things like Badge, Cueva, and Coside. Anything with green is a biological product, and that might actually be biologically based as far as a bacterial product. It could be an essential oil. It could be other products that we're notified about to see if they have some capacity to kill spores. And then the brown is actually just the spreader sticker. We wanted to see for our field trials, does that do something to spores? just by themselves. Now you can see there's a broad range of inhibition here. Starting on the left, you virtually kill all spores when the product is in contact with CLR, versus some to the right, which have no effects on germination. So overall, systemics work great, Preaxor works great, the coppers work great. As far as actually seeing inhibition of germination of spores, things that are registered for use, double nickel, serenade, Cafidoc. I mean, these all actually have the capacity to inhibit spore germination when there is direct contact. Overall, to note, it's important to know, obviously follow the law. It's the label, it's the law. What is the active ingredient? It's important to know the mode of action, because some of these products don't claim to have a direct effect on the pathogen itself. Some actually trigger plant defense mechanisms. So in that case, you would not see germination inhibition, which are some of the products off to the right of this graph. But overall, it gives us a good first look, a way to prioritize what we study in the field. And again, we're always getting calls for potential products, and this is the best, quickest way to see if it might have some effect on the disease cycle. Okay, to move into the field testing. In 2021, we were successful at controlling CLR in the field with a rotation of badge, serenade, and double nickel. So this, moving forward to 2022, was our field control. We also had the opportunity to have no spray whatsoever, so a control of no treatment. And also, again, what does water and just the surfactant widespread do out in the field, since that is mixed with each product? We also looked at Problad, Cafidoc, Preaxor, to help support the IR4 program, some of the systemics. We also looked at a different copper for rotation in still, which included the serenade and double nickel. Now, our team actually sprayed every time for the application. And for certain treatments, there was the combination of either a backpack sprayer being used or a mist blower to see if one provided better coverage. Okay, so this is a map showing our field trial setup in Holualoa. About 1,300 to 1,400 feet in elevation, set up in 3 blocks. And each color represents one of the 13 treatments that was sprayed in the field. We started monitoring in May, and the first application of these products were applied at the end of May, with then a second application approximately 8 to 9 weeks later. We started taking incidence data right away, overall with good plant health, pruning, fertilization. The percent incidence of coffee leaf rust remained rather low to start. And for that reason, instead of just looking at the incidence of random branches, we actually looked at the incidence overall for each tree. Okay, so as a snapshot in time, here's the end of August. The various products, and these are the average of the 3 blocks of all the trees and the data, the incidence data collected. And you can see when the scale is stretched out, you do see differences. The lowest ones, again, being all of the systemics, Preaxor. But if you note, the scale rating at this stage, week 13 into the trial, the incidence is still, for the most part, for all of them, well below 5%. Okay, another way of looking at it, we wanted to see if there was variability in the blocks. So where the previous slide was the average of all 3 blocks, the trees there in all 3, this slide actually shows the separation of each block. And in many cases, there was similarity in all the blocks, but you can see there's definitely variability depending on where you are in the field. So not every location, and this really speaks to the microclimate available, whether that be the airflow or perhaps the shading, but you can see that in some instances, there's definitely a hot spot to pay attention to. But again, in most cases, well beneath that 5% incidence rating. Okay, so to try to summarize the field data over the season, this graph is showing you from week 13 into week 23. So that's the end of August through November into the harvest season. The various colored lines indicating the treatments with the various controls, Preaxor, the systemics, the combination of the copper and the Serenade and double nickel, no treatment, and also just water and widespread. And the 5% line, which is indicated by that dashed black line, again, shows you how far into the season we were able to get very good control when the incidence level at the start of the season was very low and there was good management practices and at least a couple sprays. Overall, incidence was quite low, but it did separate out into a few different groups, which I've tried to indicate with the red star, the green pound sign, and the purple up at top. Definitely the systemics and Preaxor limited incidence throughout the entire season. You had that grouping in the center, which, believe it or not, all of the controls, including a no spray control, remained quite low. And we definitely believe that because management for incidence, the disease was very low throughout, cutting down on the inoculum load, we were able to get good control no matter what treatment was being used in this case. Okay, so I just wanted to give the reminder of the various fungicides and what they do. So the left picture showing those blue spots, the fungicides being applied, and then what happens when it's the various products. So the first picture you see is the contact, that would be the biologicals. Technically oxidate, where the spot hits, and that's copper, the copper products as well, you will get some slight spread, but it will not continue to spread throughout. Okay, so the drop, depending on droplet size, you'll get a little bit of a spread, but it won't protect anything more. So if it only hits the upper leaf surface, you'll have no protection in the lower leaf surface. Okay, now you're looking, the third picture is the translaminar. You will see movement of the product. If you reach the upper surface, it will translocate to the lower leaf surface, but it will not go systemic, and it will offer no protection for any new production of leaves. The third case are the systemics on the right. No matter where you apply the product, it is taken up in the vascular system and it moves throughout. It will protect new leaves and it will offer good protection throughout the entire plant. Okay, so good spray coverage is key. We've learned that not only in the field, but especially in laboratory experiments. So this is again to show you those chlorotic spots from the upper surface of the leaf on the left, the first picture. If the coppers, the biologicals, even oxidate, if you're trying to kill Hamakua vastatrix, CLR, you will have no effect on anything going on if you just cover this left picture, okay? The underside is where the active spore production is. So those biologicals, the coppers, need to reach this area. In this case, for this advanced stage of disease, you will kill some of these spores, but it won't actually do anything to the fungus living inside, which will continue to produce more spores over time. Now the translaminar, if you sprayed the translaminar Preaxor onto the leaf on the left, it will actually go from the upper leaf surface to the lower leaf surface. And in this case, knock out the spores that you're seeing, and more than likely, most of the mycelium on the inside of that middle picture. Okay, now why a systemic will be key, a true systemic, no matter where, it will translocate in the vascular system. It will get to where it needs to go. And so we're looking forward to the work with the IR4 program for the amended labels. Now, just to show you that even with our application of a potted plant, this is a good example on the right side, we sprayed copper. We thought we got good coverage. We let the product dry and then immediately inoculated the plant with CLR. And you can see here within about 3 weeks or so, 3 to 4 weeks, we started seeing these yellow spores. Well, if you zoom in and what you see, we did not get good coverage for no sticker spreader. Although some of the droplets moved, obviously it didn't protect all areas. So when fresh spores landed, they were able to germinate, enter the stomates, and start the disease. Okay, so to move into variety testing, which we've been doing a lot of, this is our overall methodology. In this case, it's a collaboration with HARC in receiving leaves from Oahu. 4 main steps. We prep the inoculum, we get fresh spores and get them ready to go. We receive the leaves from HARC, and in this case, a big shout out to Saya and Ming Lee, and especially Dr. Chifumi Nagai for having this type of research program, the breeding programs decades ago, that we can now actually test here in Hawaii, looking for resistance to CLR. In the third case, we inoculate these leaves after making leaf discs. And then the fourth step is data collection. We actually look for the incidence and severity 2, 3, and 4 weeks post-inoculation, whether that being the chlorosis and the sporulation caused by coffee leaf rust itself. We also note if there's natural necrosis just caused by tissue decay, and we use a severity rating that is found in the literature. Okay, this is just to show the left side what these leaf disc assays look like. Again, we receive leaves or we get leaves. This is the same procedure if we're testing on island. We have these Petri dishes lined with an absorbent material because you need high moisture, presence of some water on the leaves, but not too much. So we protect the leaves with by sitting on an insect screen, and spores are inoculated onto these discs. We have a giant chamber that we can optimize the conditions for disease, and we inoculate and watch and take data over time. And so this is to show you just kind of the zoom in of susceptible control. So we always have something that will produce these spots and spores at a certain period of time, in this case, 3 weeks after inoculation. And we compare that then to these different varieties that we're testing. Okay, so this is a great team effort. And these are the results up to December of last year. And again, on this y-axis, it's the percentage of discs that sporulate. So in order to complete the life cycle, the pathogen gets in, it grows inside, it produces spores, and that would be considered one cycle. This new spore, which can now cause additional infections, either on that leaf or somewhere else. So we really want to see, well, where is it that these varieties either inhibit growth altogether or have a much reduced symptomology develop over time? And you can see this graph. There's definitely variability. The right side being almost 100% of the discs showing spore production over time. The left side showing, and a good portion of this kind of this curve of material that hardly shows spore production. And to just give a bit of a picture representation, our controls, the susceptible controls on the left, you see the spots covered in these powdery spores. The middle is what we're calling tolerant or potentially resistant. We get these spots, these visible spots. So the spores get in, they germinate, but they don't actually complete their life cycle and produce more spores, or a very reduced amount of spores are produced compared to the susceptible variety. And we even have instances, that's kind of the exciting news, where it looks like resistant material, we don't even see those chlorotic infection spots. So stay tuned for more research to come. And the final topic will be our one of our favorite ones, the natural enemies of coffee leaf rust. Overall, we've been conducting a mycoparasite survey. That just means fungi that attack the coffee leaf rust spores. And we've been to a number of farms and field sites pretty much throughout the entire island now, which varies in elevation, the amount of rainfall per month, whether it's shade, partial shade to full sun at these locations, and the basic methodology. We see these spots with spores and fungi attacking those areas. So the spores are scraped onto selective media plates. The CLR spores won't actually grow. Again, they need a living host to survive, but what grows is what is attacking it. And we're able to isolate them, purify them, actually identify them both by their DNA and what they look like under the microscope. And at this stage, we've looked at more than 500 individual lesions to show the diversity of what we can find here in this picture. We have close to 200 isolates to work with, which comprise more than 42 genera. Okay. The next stage, okay, just because we've isolated them, what does that mean? Well, can they stop either spore production or kill spores or inhibit the pathogen altogether? And we've been able to now look at some efficacy studies by inoculating the spores with isolates, going through an incubation phase, and then taking data over time, taking pictures and calculating what we're calling the severity. How much of the coffee leaf rust area are these isolates colonizing? And we can use a program called Assess to do that. And this is just an example. Again, we can use, begin with leaf discs. Our control on the upper left shows abundant spore production by coffee leaf rust, H. vistatrix. And what happens when we add a sample, which at day 0, you cannot see anything, but over each day taking pictures, and here's day 7 after inoculating these mycoparasites, they start to colonize that area, actually attacking the spores. So they look a little different, they colonize a little different. And what is this telling us? Well, just to give a brief example of those previous pictures, there's multiple species of a genus called Simplicillium, compared with Lecanicillium, and these are actually good biocontrol candidates that are found in other parts of the world, especially attacking the coffee pathogen. And over time, how well does it colonize that spot? You can see that, well, not all mycoparasite behaves in the same manner. Some within day 3 have colonized at least half of it. But long story short, in a week's time, all of these isolates are colonizing almost 100%. And now we're actually seeing, well, how detrimental is the mycoparasite to the spore themselves. So stay tuned for more information on that. So with all that being said, I know that's a lot of information. But the same message as far as what is the summary and how to combat CLR. You have to manage it continually. We continue to say there is no one silver bullet. It's an integrated approach with short and long-term strategies. Definitely, we want to avoid the introduction of new races. There's more than 50 that exist for Himalaya in the world. We have one. We want to avoid new things coming in or new things developing here. With that, I will say thank you very much. Please don't hesitate to contact me. There's my email. That's the best way to reach me. I'm happy to answer any questions. I definitely thank our funding source, and check out Andrea's website for just a plethora of information. And with that, I say thank you very much, and appreciate your time.